Hey guys, welcome back to another video. So in this video we'll be talking about DNA manipulating enzymes, manipulative enzymes. So DNA manipulative enzymes are very much important in most processes such as isolation and many other processes. So we'll talk about some of the manipulative enzymes which are involved. So there are four types as you can see portrayed in the picture. So our first type is nucleases, then there is polymerases, we have ligases and other modifying enzymes as well. So we'll talk each one of them about each one of them. So watch the video till the end and do click the like button if you like the video. So talking about the first manipulative enzyme, which is the nucleases. So basically the function of nucleases is to degrade nucleic acid by breaking the phosphodiester bond. So this is the fundamental step for breaking any nucleic acid. So because the nucleic acids are linked by phosphodiester bonds, so the main primary motive to destroy nucleic acid would be to break the phosphodiester bond in either ways, in any of the ways. So nucleases does the job of breaking the phosphodiester bonds so that it can degrade the nucleic acid molecules. Alright, so that further link one nucleic acid to the another DNA or RNA cell. So basically it degrades the nucleic acid by breaking the phosphodiester bonds. Also, there are two types of nucleases. One of them is exonuclease and the other is endonucleases. Alright, so both of them have very important functions. I hope you have heard about exo and endonucleases. So we'll talk about each one of them. So talking about exonucleases, so its function is to remove one nucleotide at a time from the end. This is very important, from the end. So where it removes, is it from the front or the end? So exonucleases, as the name suggests, it removes from the end of a DNA or a RNA strand. So as you can see in this picture, so this is, let's say, a DNA or a RNA strand. All right. So exonucleases activity happens here as well as here. So it says from the end. So it is not specifying whether it is from the 5 end or the 3 end. So both of them, we know 5 and 3 are the ends of a particular any DNA or RNA strand. So in this case, exonucleases does the job of removing one nucleotide. Just it is specified that it would remove only one nucleotide at a time from both the ends. All right, or can be from either of the ends. All right, of a DNA or RNA strand. So as you can see in this picture very clearly, this is a DNA or RNA molecule. So from this is one end, this is another end. So let's say this is the 5 end, this is the 3 end. All right. So it removes nucleotides, one nucleotide, that is very important, removes one nucleotide at a time from both the ends, and thus we get a new modified strand like this. All right. So this is the job of exonucleases. So moving on with this. So now we'll talk about some of the examples of exonucleases. So let's just study the entire exonucleases before we move to endonuclease. So talking about the first type of exonucleases, which is the BAL31, BAL31. So basically it is purified from the bacterium Alteromonas aspergillus. So this Alteromonas aspergillus is the bacterium from where we get BAL31, which is a exonuclease type of enzyme. And its function, it's very important to know the function. So it removes nucleotides from both strands of a double-stranded molecule. Alright, so it removes nucleotides from both strands of a double-stranded molecules. As you can see in this picture very clearly. So it removes nucleotides from both strands of a double-stranded molecule. So both strands means from both the ends. Alright, so this is the end, this is another end. Thus we get modified DNA like this. This is how it removes, and in the further process it will keep removing nucleotides from both the ends. Alright, so moving to the second type of exonuclease, which is the exonuclease 3. So it is purified from E. coli. So from E. coli we get exonuclease 3, and its function is to degrade just one strand of a double-stranded molecule, leaving single-stranded DNA as a product. Alright, so it degrades only one strand. So let's say this is a DNA sequence. Alright, so it will degrade only the upper strand or the lower strand, and it will degrade just one strand of a double-stranded molecule, leaving the single-stranded DNA as the product. So let's say it would degrade only one strand at a time. So from here it removes this nucleotide, this base pair, this base, and from the upper strand it removes this. So it leaves this much from the first degradation. All right. This is how it will keep removing from here this one, from here this one. So it will keep removing or degrading or removing bases from one strand of a double-stranded molecule, leaving the single-stranded DNA as the product. Whereas BAL31 removes nucleotides from both the strands. So it does not discriminate between each of the strands, whereas it removes nucleotides from both the strands, like from here as well as from here. Alright. Whereas it is not the case with exonuclease 3. Alright, so moving on with this, we move to the next. So talking about endonucleases, which is the most important point. So as you can see, on a lot of pen marks are there. So this is all for your understanding. So let's talk about the function of endonucleases. So its function is to break internal phosphodiester bonds within a DNA molecule. So it is very much similar to the exonucleases. So it does the same, but it breaks internal phosphodiester bonds within a DNA molecules. Whereas exonucleases breaks internal molecules from the ends. Like this is a DNA strand, so it takes a nucleotide sequence from here, or either from here or from both the sides. All right, at once. Whereas in this case, it will break these internal phosphodiester bonds. All right, so this is the job of endonucleases. All right, so talking about some of the examples of endonucleases would be, so the first one is S1 endonucleases. So this is purified from fungus Aspergillus oryza, and it cleaves only one strand. All right, so it does it as you can see in this picture. So talking about S1 nuclease, so as you can see the function very clearly as I have circled it. So it is purified from fungus Aspergillus oryza, and it cleaves only one strand at a time. So let's take this is a strand, or let's say this is the DNA, and it cleaves only this part. As you can see, I've wanted this one. So this has removed one of the nucleotides from here, and it degrades only one strand at a time internally. That is very important. All right. So thus it does like this. In this case, it removes this nucleotide, so it becomes leaves somewhat a single strand, one strand at a time. As you can see in this picture, this is a one strand. It removes a base pair from this side, and thus we can see a modified DNA strand. All right. So talking about another endonucleases, which is deoxyribonuclease 1, which is DNA 1. It is isolated from cow pancreas, and it cleaves both single and double-stranded molecules. Alright, so it cleaves both single and double-stranded molecules, whereas it was only for single-stranded, and it is for both single and double-stranded. Alright, so it is DNA 1 is non-specific, and it attacks DNA and internal phosphodiester bonds. So it is very much non-specific, and it can attack to any of the internal phosphodiester bonds at any positions. It is not defined. So the end result of a prolonged DNA's one action is a mixture of mononucleotides and a very short oligonucleotide. So as you can see, this is a strand again. This is pretty much similar to S1 endonuclease. So it removes base pair from here. So thus it leads to formation of this again. This is also same in this. Let's take a double-stranded molecule. So it removes or what you say, it removes bases from both these strands. So it does not discriminate between single or double strand. It removes from both these strands, which was not the case from this. So it used to remove singly or cleaves only one strand at a time, whereas it would leave multiple strands or double strands at a time. So it would take away both the base pairs in this case at a time, so that a new modified DNA can be formed. All right. And talking about the third endonuclease enzyme, which is restriction endonuclease enzymes, it cleaves double-strand DNA only at specific recognition sites. Alright, so this is similar to deoxyribonucleases, but just one difference, that it is specific, whereas this is non-specific. Alright, so it is specific at this region, so it removes base pairs at this region at double-stranded, and it degrades it degrades or cleaves double strands and not single strands. It is very similar to deoxyribose 1 and it is very much specific, whereas deoxyribose is not specific. So that's it for my video. So I'll be making another video for completion of this entire chapter of enzymes, DNA manipulating enzymes. So thank you for watching this video. Stay tuned for more.